So, hello everyone. I first like to thank the organizers, HFSP, Guntram Bauer, and all the other organizations, institutions involved in bringing this fantastic conference together. I'm a neuroscientist, and it's a great pleasure to talk to an audience with such a diversity of expertise and backgrounds. And I realized, putting this talk together, that my title is a bit clunky and technical in this regard. What my talk will be about is how the brain, or parts of the brain, adapts to heat to help us to become more tolerant, more resilient to heat. So, my lab is interested in temperature and how temperature interacts with the body, and we use the model system, the mouse, as our main research object. Let me see, how do I, yeah, good. So how do we detect temperature? For example, if you touch a hot plate, if you jump into a cold lake, what is it that allows you to detect these temperatures? Well, that is quite well established by now. There are little molecular thermometers called TRP, or transient receptor potential channels. And 2 years ago, the Nobel Prize was given to my former postdoctoral advisor, David Julius and Ardem Pataputian, who identified and started to characterize those channels with many, many other groups in the world by now. So you can visualize those molecules as little thermometers that sit in the cell membrane, and there are diverse versions of these, and they respond to different temperature ranges. And in the context of the cell, temperature makes drives the conformational change. This channel protein opens up, ions flow into the cell, and this is then the signal for the cell that a certain temperature has been reached. Now, in the context of our sensory system, these channels detecting warm, cold, hot temperature are expressed in sensory nerve fibers that innervate the skin and other body parts. So they create, upon a temperature stimulus, an electric signal that is then relayed along the spinal cord to the brain, where then we feel or process temperature information based on these inputs. Now, okay, so peripheral temperature detection is quite well established, but how about temperature regulation on a cellular physiological basis? There, much less is known. How this temperature information is integrated to really allow us to maintain a stable core temperature. And that's why this is so important. We all know our body temperature fluctuates slightly around our core temperature of 37 degrees. Our model organism that we use in the lab, my team uses the mouse, the same thing, 37 degrees. And what is quite astonishing is we can keep this temperature stable despite very diverse external temperatures, and also despite very diverse and fluctuating heat generation processes like exercise or metabolism. So how does this work? Well, we in the brain, or parts of the brain, have something like a thermostat. A computational unit that behaves like a thermostat, and that allows us to keep and maintain a stable core temperature. So how does this thermostat work? That's an essential question that we like to address with many other labs in the world. What happens downstream of that thermostat is also quite well established, and I think we all can appreciate now walking down in the hot environment outside. Well, first of all, the thermostat can trigger heating and cooling. Different organs are engaged in the body. I will here focus only on cooling now, because that's what this conference is about, global warming as part of it, as at least one aspect, and therefore I will focus on mechanisms of cooling. So cooling mechanisms that we have at our disposal is, for example, sweating, blood flow direction to the skin to dissipate heat to the environment. We all know this. When we blush, when we run, when we exercise, we turn red because we dissipate heat to the surrounding. We can reduce heart rates. Our animal model organisms do this if they are exposed for a long time to heat. We can lower metabolism, and of course there are behavioral adaptations, like moving away from the hot source and going into a shade, for example. So the mouse, our favorite, preferred at this point at least, model organism, has similar capabilities. It can direct blood flow to the surface. It mostly uses the tail for this because that's the only non-furry part. It can reduce the heart rate. It can switch off the brown adipose tissue. So the mouse generates a lot of heat by brown adipose tissue, and it also has the ability to regulate by behavior. It can move to cooler places, into the shade, or into underground burrows. Now, who is orchestrating all these diverse responses? Now, there is a region deep in the brain called the preoptic area, and this region, as part of the hypothalamus, really resides at the very bottom of the brain. And it's highly conserved. It's actually, from the cell molecular architecture, similar to that also in humans and most mammals, as far as we know. It's a conserved brain region all the way down here. So here's the cortical region. So we are looking at this tiny region down at the bottom of the brain. It serves, so to speak, as a thermostat, and directs temperature mechanisms, and it does so, needs feedback information, of course, and it integrates temperature information from the temperature sensors that I just mentioned to you. Now, the question for us really is, how does it work? How does a set of neurons can do this? And please, I put thermostat in quotes. Thermostat static implies that a thermostat is something that is very, very stable, but in our context, actually, it's quite adaptive and quite plastic. That's why this analogy doesn't hold true from a pure engineering point of view. Now, a couple of years ago, us and others identified what I call here thermostat neurons. And I just want to show you one example what these thermostat neurons do when we artificially manipulate them and activate them. So in this context, what we did, we introduced, Hong Wang, a postdoc in my lab, former postdoc in my lab, introduced with a genetic trick, a switch into these neurons. And this switch we can toggle between on and off, so we can turn these cells on and off to our liking remotely. And we can ask when we turn these neurons on, what does that do to body temperature? So just a little bit technicality here, bear with me. So these neurons, these thermostat neurons, actually at baseline, under normal environmental conditions, which are actually quite 22 degrees, like we probably have here, is actually quite cold for mice. And these neurons are not very active. So you see they communicate by spikes, action potential, and you see just one spike, otherwise it's a flat line. Now, when we introduce this engineered receptor into these cells and engage it with a drug that we inject into the mice that we can supply to the animals, what it does, it makes these neurons fire. So they become very activated. And the simple question we ask is, what happens to body temperature of mice when we do this in this discrete set of neurons in the brain? And to analyze this, we have various means. We use here, and I show you here, infrared imaging. So this is an image from the top into the cage of the mouse. The mouse can freely move around. You can see the body is nice and warm. And you can also note that the tail it uses to thermoregulate is actually under these conditions, baseline conditions, cold. You can hardly see it. So it's almost like environmental temperature, 22 degrees. And also the ears is sort of like the exposed areas that are a little bit cooler. So now what happens if I turn on these neurons? So now it's still the baseline conditions. Just to show you again, in the moving, the animal moves around. And now we switch these cells on. And you can see immediately the tail lights up. The mouse dissipates heat to the surroundings so much that it loses heat and heats up the bedding, with the effect that the mouse cools down. It cools down dramatically. It loses 10 degrees of body temperature, from 37 to 27. And actually, the nice thing is with mice, if the switch is turned off, they're fine. They come out of this torpor-like low energy, low temperature stage. So what I've shown you is, and a lot of other experiments at the time, we could show that certain cells, certain neurons in the body are very, very tuned to, or very much important for regulating body temperature. And other groups have also contributed this, identified different cell types in this small brain region. Now, this is a very artificial experiment, obviously. These neurons are not very active. Normally, we artificially activate them. Here we ask the question, what naturally does activate these neurons? And we reasoned heat might play a role here, heat stimulation. And particularly, we looked at heat acclimation, so exposing mice, our model organism, to warm temperatures for a long time. So we asked, what would happen to these thermostat neurons if we do this? And two postdocs in the lab, Sarah Nencini and Wojciech Ambrosiak, they embarked on this from different angles. And we acclimatized these animals for up to 30 days at 36 degrees. So normal vivarium temperatures is around 22 degrees. I think most labs that work with animals have this type of temperature. And we exposed them to different temperatures. And we asked these thermostat neurons what would happen. And as you can see here, quite dramatically, the neurons become very, very active. Not after the first day, second day, but then they really become more and more active. They adapt in this cellular fashion and become very active. We can quantify this. And at the same time, what Sarah did, she asked, can we measure the physiological response of the animals, what they would do, or what happens to them when they acclimatize? So, well, first, heat acclimation induces activity in these cells. So when mice are acclimatized at the same time, they become more heat tolerant. And there's various ways to measure this. We used a so-called heat challenge or heat endurance assay, where we telemetrically monitor the body temperature. And we do this after exposing the mice to this heat acclimation protocol or not, keeping baseline. And what you can see is here, very simply, so here's the time they are exposed to this really strong heat challenge. For mouse, 39 degrees is a really, really strong challenge. And so here's the cutoff. This is the end of the assay. It's sort of a life and death thing. We rescue these animals at this point and take them out of the assay. And you can see, because 41.5 degrees is sort of the limit that they can tolerate. And you can see mice that are not acclimatized don't do very well. But if we acclimatize these animals for a long time, they actually can go beyond the circadian cycle of 24 hours. We have to stop the assay then. So they probably could go on even longer. We can again quantify this. Here, that's what we call the endurance time after at this strong heat challenge. Now, what I've shown you is mice can acclimatize and gain heat tolerance, as do we, probably by other means to be shown. But so what I've shown you is that the activity of these neurons goes up. At the same time, mice become heat tolerant. And the obvious question is it merely a correlation, or is there a causal consequence? Is it required that these neurons become more active to make these mice more heat tolerant? And to address this, we again used a similar genetic trick, switch, but in the opposite direction. Now we switch these neurons off. So as I showed you, these neurons become very active by heat acclimation. So we acclimatized the animals, we put in an off switch and asked what would happen if we then silence the neurons, what would happen to heat tolerance. Now here, this is just one slide of real data, so to speak. The other traces were sort of illustrating things. So you can see here these acclimatized neurons. We can very nicely switch completely off when we introduce this genetic engineered receptor and quantify this. And then again, we acclimatize these animals and ask how they would do in the heat challenge assay when we switch these neurons off. And the result is very, very clear and very shown here. So again, the control conditions, mice can cope with heat quite nicely. They don't reach this cutoff of 41.5 degrees. But the same mice where we then switch these neurons off, they fail very, very early on. And this we can quantify here as well. So what we can say is that the activity of these thermostat neurons is required for the mice to display heat tolerance that they gain upon acclimation. So we went one step further and asked, can we do this artificially? Can we acclimatize mice without ever having been exposed to warm temperatures. And here we used another genetic trick that is quite well established by now in the neuroscience field called optogenetics. It allows us optically with the light stimulus to activate neurons to our liking and to the length we choose. And we did this, we artificially activated these neurons for many days and asked how would they do in this heat tolerance assay without ever having been exposed to heat before. And you can see if we do this for 1 day, they don't do very well. But then after 2 days, one can see already a trend, and it becomes significant after 3 days. So 3 days of optic activation of these neurons rhythmically, as they would naturally if they are exposed to heat, allows these animals to become heat acclimatized, so to speak. And this allows us now the conclusion with many other experiments we did, and I don't want to go into detail here, we can of course discuss later on, that the activity of these neurons is not only required, but also sufficient, at least for in this type of assay. One can monitor heat tolerance, of course, in many different assays, and these heat challenge assays to become heat tolerant. So with this, I like to summarize what I've shown to you. So temperature is detected in mammalian species like us or mice via trip channels. The temperature information is relayed to the cortical area, that is sort of then the sensation of heat or cold, but also to this evolutionary, highly conserved region in the hypothalamus, the preoptic area, and that is then autonomic responses mostly that help us to thermoregulate. And if we expose animals for a long time to a warm, hot environment, these neurons change plastically, and we have also started to uncover what mechanism, molecular mechanism, drives this pacemaker, this activity change. And this activity change is required then for animals to become heat tolerant. So with this, I like to acknowledge funding sources, ERC, German Research Foundation. I'm actually affiliated also with EMBL via a construct in Heidelberg, the MMPU, Molecular Medical Partnership Unit. And I also acknowledge HFSP, Wojciech Ambrosiak, one of the postdocs who generated lots of these data, of which I was only able to show you a glimpse, was also an HFSP fellow. And I'd like to thank you for your attention.